I told you, I'm not angry. I'm overstimulated!
Good day, E-PASAR students. Welcome to another biochemistry review with your favorite team of tutors. Today, we'll talk about something spiral, stable, and super important. What is it? The alpha helix, a type of secondary protein structure. So first, let's start by understanding what secondary structure actually means. So secondary structure is the localized folding of polypeptide chains. This is thanks to the hydrogen bonding between the backbone of the atoms of amino acids. Now, there are two main types of polypeptide chains. First is the alpha helix, next is the beta plated sheet. But today, we're focusing on the first one. Now, let's zoom in on the alpha helix. As you can see, it is a right-handed spiral, kind of like an old school telephone cord or a spring. It's tightly coiled with a 3.6 amino acids per turn, a rise of 1.5A per amino acid, an intrachain hydrogen bonding between the NH and CO groups of amino acids. And it has four residue apart. Each hydrogen bond is weak, but the large number involved gives a very stable structure. And don't forget, the R groups stick out. They are like the arms on the outside of the spiral. This keeps the inside very clear. And there are very many variations of an alpha helix. So let's talk about them. First is the presence of proline. This is basically what disrupts the alpha helix because its side chain loops back to the backbone. This locks the phi angle and removes the amide hydrogen needed for bonding. This causes a kink or break-in the helix. Next is the presence of adjacent similarly charged groups. So if multiple residues with the same charge like several lysines or glutamates are close together, they repel each other. This electrostatic repulsion destabilizes the helical structure. And lastly, we have the presence of adjacent bulky groups. These are amino acids with large side chains like tryptophan. Next to each other can create steric hindrances. And this crowding makes it very difficult for the helix to coil properly, leading to instability. Now here's a short animation of the alpha helix, which shows you its hydrogen bonding. And you can notice from the video all the things that we have already explained, like its interchain hydrogen bonding with the NH and more. So that's our first look at the alpha helix. Now let's explore what keeps this structure so strong with Febrienne.
So thank you, Danielle, for that introduction. So now, E-PASAR students, now that we know what an alpha helix looks like, let's ask a key question. Why doesn't the alpha helix fall apart? Well, the answer lies in several stabilizing forces that work in harmony to maintain this structure. Without this, the helix would unravel due to thermal motion and solvent interactions. So let's break them down one by one. So our first stabilizing force is the hydrogen bonds. So these are the primary stabilizing force of the alpha helix. So each carbonyl oxygen from one amino acid forms a hydrogen bond with the amide hydrogen of another amino acid 4 residues ahead. So this repeated pattern of bonding produces a rigid, spiral staircase held tightly together. So these bonds run parallel to the helical axis, giving the helix a remarkable mechanical strength and resistance to unraveling. In fact, there are about 3.6 amino acid residues per turn of the helix, so with each hydrogen bond spaced approximately 1.5 angstrom along the helix axis, creating a tightly packed and compact structure. So next stabilizing force is the dipole moment. So the alpha helix has a macrodipole because of the aligned peptide bonds all oriented in the same direction. So the N-terminus accumulates a partial positive charge, while the C-terminus accumulates a partial negative charge. So this dipole affects how the helix interacts with other molecules. For example, negatively charged residues like glutamate are often found near the N-terminal end, while the positively charged ones like glycine prefer the C-terminal end to stabilize the charge imbalance. So in membrane proteins, this dipole can even influence how helices insert into the lipid bilayer or orient themselves within a folded domain. So the next stabilizing force is the van der Waals interactions. So let's zoom in on the side chains of amino acids in an alpha helix. So notice how they all point outward from the central helix. So when several alpha helices are close together, especially in the tightly packed hydrophobic core of a protein, these side chains can fit together snugly, almost like puzzle pieces. So this tight packing allows van der Waals forces to come into play. So these are weak, short-range forces caused by tiny shifts in the positions of electrons and atoms. So when two non-polar side chains get very close, their electron clouds can temporarily synchronize, creating a transient dipoles that attract each other. So even though each attraction is small, the sheer number of these interactions adds up, just like thousands of tiny Velcro hooks sticking together, so they collectively stabilize the protein structure. So our last stabilizing force is the hydrophobic interactions. So hydrophobic residues such as valine, leucine, isoleucine, phenylalanine, and methionine are non-polar amino acids that tend to avoid contact with water. So in protein folding, these residues hide inside the protein structure, while polar or charged residues face the aqueous environment. This behavior is known as the hydrophobic effect, and it's a major driving force in maintaining protein stability. So in globular or water-soluble proteins like enzymes or plasma proteins, alpha helices tend to fold so that hydrophobic residues are packed tightly in the protein's interior, while hydrophilic residues face the watery surroundings. So in membrane proteins such as receptors and channels, alpha helices often span the lipid bilayer, embedding themselves like pins through the membrane. So in this case, the hydrophobic side chains face the fatty acid tails of the membrane lipids, while any exposed hydrophilic regions may face the inside of channels or the aqueous cytoplasm. But not all amino acids play nicely in a helix. Some disrupt it completely, and we call them helix breakers. So let's explore the two main ones. So first we have the proline. So proline is often called a helix breaker. First, because of the hydrogen bonding disruption. Most amino acids can form hydrogen bonds that stabilize the alpha helix, but proline lacks an amide hydrogen on its nitrogen atom, which means it cannot donate a hydrogen for hydrogen bonding. Thus, this breaks the chain of stabilizing hydrogen bonds essential to keeping the helix intact. Second is the rigid ring structure. So proline's side chain loops back and bonds to its own nitrogen, forming a 5-membered ring. So this structure locks the backbone angle in place, limiting the flexibility needed for the helix to twist smoothly. Third is because it bends instead of coils. So because of its rigid and bulky shape, proline causes a kink or sharp bend when inserted into an alpha helix. That's why it's rarely found in the middle of a helix. Instead, you'll often see it at the start or end of a helix, in turns or loops where bends are actually useful for the protein's 3D shape. So our second helix breaker is the glycine. So glycine is the smallest and most flexible amino acid. Its side chain is just a single hydrogen atom. So this gives glycine maximum rotational freedom, unlike bulkier amino acids such as alanine. So because of this high flexibility, glycine cannot maintain the rigid geometry required for a stable alpha helix structure. So why is this important? Well, the alpha helix is stabilized by hydrogen bonds that form between the carbon double bonded to oxygen group of one amino acid and the nitrogen bonded to hydrogen group of another amino acid that is 4 positions ahead in the sequence. So this stabilizing pattern requires consistent spacing and fixed bond angles throughout the helix. So when glycine is present in the sequence, its excessive flexibility disrupts this uniform hydrogen bonding pattern. So as a result, the helix becomes unstable and may develop a kink or begin to unwind. So now, E-PASAR students, when predicting or modeling protein structures, remember to look for the alanine, glutamate, and leucine, which are great helix formers. Hydrogen bonds, dipole effects, van der Waals, hydrophobicity, which are factors that stabilize the structure. And remember to always watch out for proline and glycine, which are common helix breakers. Understanding these forces helps predict folding patterns, interpret protein functions, and even design synthetic proteins or drugs. So up next is Naysa, where she will guide us through real-life examples of alpha helices in biological systems.
Thanks, Febrienne. Now that we know the structure and stabilizers, let's ask, where do we actually find alpha helices in real life? First, in hair, skin, and nails, alpha helices are part of alpha keratin. Second, in membrane proteins, helices span the lipid bilayer to form channels or pumps. And third, in DNA-binding proteins, helices fit into the DNA grooves to help with gene regulation. Alpha helices are everywhere in the protein world because they're compact, stable, and versatile. Now let's review what we've covered today. So here are the alpha helix key points. First, they are secondary protein structure. Second, they are right-handed coil and have 3.6 residues per turn. They are also stabilized by I to I+4 hydrogen bonds, and proline and glycine disrupt helix formation. They are also found in keratin, channels, and DNA-binding proteins. And that's your quick but solid spiral dive into the alpha helix. Here at E-PASAR, we believe understanding the why behind protein folding can help you master more advanced concepts in biochemistry and pass with flying colors. Thank you for learning with us. We are your E-PASAR review team. See you in the next session.